Okay, so this is the review for exam 2, and so I'm just going to go through basically the lectures from chapters 6, 7, 8, and 9, and sort of highlight what you guys need to see or learn. All right, so this is chapter 6. I'm just going to highlight the things that you guys need to know. Since I've moved the screen, it's going to pause it. Okay. Okay, so chapter 6 is tour of the cell. And by the way, there's probably going to be some construction in the background. So I apologize for that, but it is what it is. I really don't have anywhere else to do this recording because of the software. Anyway, so for the exam, you're going to need to know who Robert Hooke is. Remember, I told you guys you don't need to know dates. You just need to know people. So we already talked about Robert Hooke. We talked about him in chapter 1. So you should already know this. It should be a review. Remember, he used a compound microscope, and that had 2 lenses, an ocular, which is next to your eye. That's what ocular means. And then an objective lens that is closest to the object or specimen that you're looking at. So the objective lens is here, the ocular lens is here, and then the magnification's combined. So if this is let's say this is times 10, this one might also be times 10. And so that magnification would be 10 times 10, which would be 100. A 100x. Okay, so I don't care if you know about a condenser or not. It's not going to be on the test. But you do need to know ocular because that's a specific lens, and if you do any sort of science or medicine, you're going to need to know that. The objective lens, that's the lenses that are close to objective. Usually there's 4 of these, and you can rotate them. So usually they're 4x, 10x, 40x, and then 100x. And if you guys are in school, in the lab, you would be able to mess around with these microscopes. The image is inverted here and then reverted back through the eyepiece. So you see it upright. Remember, there's a difference between magnification and resolution. Oh, yeah. So magnification is the amount of the increase that you can have. If you ever use like a magnifying glass, you know that you can magnify the heck out of stuff, but it gets blurry, and that means you lose the resolution. So remember, just because you make it bigger doesn't mean that you can still see it clearly. Resolution is like a high-def TV versus an old TV. Resolution is defined by the distance that you can distinguish 2 points. So, you know, if I wrote these 2 dots here and they're separate, but if you walked away, just walk away from your screen, at the point wherever it looks like 1 dot, you can't distinguish the 2, then you've lost resolution. Then the resolution of a light microscope is limited by the wavelength of light. I'm going to ask you that on the test. You need to know that. It's super important. I don't care if you know this or this, but you should know that the maximum magnification is about 1000x. That means 100x. That's why all good microscopes, their objective lens is 100x. Their ocular lens is 10. 10 times 100 is 1000. So you get 1000x magnification. That's the maximum that's possible. I will ask you what the maximum magnification while maintaining resolution is the same. All right, so electron electron mic.
Yeah. That's going to be the floor. It's going to be the walls, and that's going to be one, like the main wall, carrying the look at the angle. Yeah.
All right. So sorry for the interruption. Electron microscope resolution is inversely squared to the wavelength. So that means shorter wavelengths get bigger resolution. And electron beams have shorter wavelengths. And so it's about 1,000 times less wavelength than it is for light, and because of that, we can do about 1,000 times more magnification. So 1,000 times 1,000 is 1,000,000. 1,000,000x is the maximum magnification that you can do using an electron microscope. And then there are other microscopes that we can resolve further, like atomic force microscopes. You don't need to really know that, but I'm just telling you they're super expensive, $10 million. There's 4 of them at the Biodesign Center at ASU, and that whole building was built around those microscopes. So this table just shows you what you can see with your naked eye, all the way down to like small frog eggs. Then with a light microscope, you can see things down to mitochondria, some of the bigger organelles in the cell. And then beyond that, you're going to need an electron microscope to see things like bacteria and viruses. So for COVID, like the images that you see in COVID, you know, I might ask you, could you use a light microscope to see COVID-19? The answer is no, because you cannot see viruses with a light microscope. They're too small. You would need to have an electron microscope. And that's what these images of COVID you see are. Okay. So remember, there's 2 kinds of electron microscopes, transmitting and scanning. The transmitting one transmits so you can see the inside. A good way to tell this is a 2-dimensional image, 2-dimensional image. And then this is focused by electromagnets rather than light focal points that we do in light microscopy. Scanning is on the outside, so this is the same cells that line your small intestine. This is the cilia that moves your food through there, and you can only see the surface of specimens with the scanning. With the transmitting, you can't see that. Cell fractionation is a process that scientists use to separate out things based on their molecular mass. So we might spin it at 800 times G for 10 minutes, and you'll get this pellet. If you were in class, we would do this for real. You get a pellet, and on the top, it's called the supernatant. So you need to know those words. You need to know supernatant. You need to know pellet. Homogenate just means that you mix it till it's homogeneous. That means it's has a consistent mixture. This is what they do with cow's milk, so that it mixes the fats in with the proteins and things that are not water soluble, so that it doesn't have, like raw cow's milk has fat on the top of it. What you buy from the store, they've mixed that together so that it's all equally distributed. And then a centrifuge is something that just spins something really fast. And if I put you on the end of a string and spun you around, it would increase your weight. And you could probably tell by holding on a rope that you have to hold it tighter and tighter because your mass is increasing. That's what we're doing here. So this pellet, real heavy things are going to come out. And then we take the supernatant off, just the liquid. We put it in a new centrifuge tube, spin it even faster, even longer. And then we're going to get another pellet and a supernatant. And in fact, every time you do a centrifugation step, you get a pellet and a supernatant. And by this, we're going to get smaller and smaller things come out in the pellet. And we're basically separating them so we can study what these organelles' functions are. And this is how scientists figured out what the mitochondria do and all that other stuff. So we've already talked about prokaryotes. You guys know prokaryotic cells are bacteria and archaeobacteria. They don't have a nucleus. They have an area called a nucleoid. They have ribosomes. Remember, those make protein. They have a plasma membrane, a phospholipid bilayer, just like our cells. They also have a cell wall, and some of them have a capsule, which is like a goopy exterior. Usually, this is made out of glucosaminoglycans. We call them GAGs for short. You don't need to know that, but it helps evade other organisms in the immune system, and we'll talk about that when we talk about pneumonia, different strains of pneumonia. And then this is an electron micrograph. What kind of microscope took this? Well, it's transmitting because you can see the inside of the cell. If it wasn't, if you couldn't see the inside of the cell, only the outside, it would be scheming. Remember, no nucleus, that's what prokaryote means, and then it doesn't have any organelles. Eukaryotic cells are different. Those are all the other kingdoms we talked about in chapter 1. They have a nucleus and they have membrane-bound organelles, and we're going to talk about those organelles in this chapter. We briefly covered this in lecture, but just remember that there's a upper limit of how big cells can be, and there's a lower limit of how small cells can be. And I might ask you why on the test. I also might ask you why they need internal membranes. Like, why do we have organelles? Well, we can have different proteins in different spaces, right? We can have different lipids in different spaces. We can prevent certain molecules from interfering. So like a DNase would chop up DNA, and that would be in the lysosome. But you wouldn't want that in the nucleus because it would chop up your own DNA. And it also provides localized environments. Maybe the pH of the lysosome is 5, where the rest of the cell is 7.3. So it's kind of like your house. If you didn't have walls around your house, you couldn't have it at a different temperature than the outside. All right, so for the nucleus, you just need to memorize all this stuff. The nuclear lamina is proteins. We talked about this when we talked about the cytoskeletal components. So that's what gives the nucleus its shape. And then nuclear pores are these gated channels that allow certain things in and out of the nucleus. Remember, I might ask you, does the nucleus have 1 or 2 membranes? You should be able to tell me it has 2. The DNA is made out of chromatin. We talked a little bit about that in the lecture, and that's DNA and proteins that make up, these proteins are called histones. Together, DNA and proteins make chromatin. Long strands of chromatin make up chromosomes, which you're familiar with. Different organisms have different numbers of chromosomes. We have 46 pairs, 23 you got from mom, 23 from dad. Chimpanzees have 48. Fruit flies have 8. Elephants 56. Woolly mammoths 58. We talked a little bit about how we can clone woolly mammoths and elephants, or at least some Japanese scientists are trying to do that. There may be a technical issue because we have different numbers of chromosomes. And then, pineapple has 60. So the point of this is just because you have more chromosomes doesn't mean that you're a more complex organism. I like to think I can outthink a pineapple. All right, so a nucleolus, that's an area where ribosomal RNA is made within the nucleus. It's just a dark spot, and that makes the components of ribosomes. So this is the RNA. Remember, ribosomes are made up of RNA and protein together. So that RNA is called ribosomal RNA.
Yeah, storage unit.
So we talked about ribosomes. There's 2 kinds. We have free and bound ribosomes. Remember, the free ones are in the cytoplasm, and they make things for inside the cell. The bound ones are attached to the endoplasmic reticulum, and those make things that are going to be exported from the cell. We talked about that in lecture. Remember, the things that move between the different organelles are called vesicles. They're kind of like a UPS truck. And these things are, so the nucleus and endoplasmic reticulum are directly connected, and then the Golgi is off by itself. These things modify proteins, so we can add lipids, and we call those lipoproteins. We can add sugars, we call those glycoproteins. And all that's done either in the Golgi or the endoplasmic reticulum. Lysosomes are the stomach of the cell. Vacuoles, these are only found in plants. We didn't really talk much about it, so you don't worry about this. All right, we talked about the ER. Make sure that you guys know the difference between the rough and the smooth. This is all of the functions of the smooth, so make sure you know that. I'm going to ask you that on the test. Make sure you know the functions of the rough. All that's in here, and that's what I'm going to ask you on the test. The Golgi, remember, it has a cis and a trans side, and so make sure that you keep those straight. I might ask you, or I might put a drawing of a Golgi and ask you which one is cis and trans. The lysosome, remember, it's the stomach of the cell, and it breaks down carbohydrates, proteins, lipids, and nucleic acids. And so it's kind of like the 4 horsemen of the apocalypse. And we talked a little bit about how your cells can undergo apoptosis if they're really damaged, like when you get a sunburn. And the lysosome actually opens up and destroys the cell. So eating yourself is called autophagy. That's when you recycle things within your cell. And then you can also do phagocytosis, is when you bring in solids and then fuse that with the lysosome, because you really don't want to bring in a virus or a bacteria from outside and give it a safe place to live. You want to digest it in your cellular stomach, your lysosome, to destroy it so it can't do anything bad. And then we talked about apoptosis. The lysosome opens up and destroys the cell. Let's say if the DNA is super damaged from you laying out in the sun. All right, mitochondria. We talked about that. They're the powerhouse of the cell. We talked about this ad nauseum in chapter 9. So make sure you guys know the different parts of the mitochondria. What kind of electron micrograph is this? You should be able to answer that. It's because you can see inside, it's transmitting electron microscope. Chloroplasts, we're going to talk about this in depth in chapter 10 for photosynthesis. Just remember, these are found in plants and algae. They have their own DNA. Mitochondria also have their own DNA. So I might ask you, where's DNA found in a plant cell? Well, remember, plants have mitochondria. They also have chloroplasts, and they have nuclear DNA. So there's 3 places in plants where DNA is found. In humans, like you, there's only 2. And then remember, all of your mitochondria DNA came from your mother. We talked about stroma and the thylakoids. We also talked about the interior of this is called the matrix.
Right here, the 4 years.
Make sure you guys know the cytoskeleton. It's to do support, but I'm going to just skip ahead and tell you, you should know, you don't need to know the structure, and you don't need to know its diameter, but you do need to know what its subunits are, and you need to know what its functions are. So I might ask you, what's in charge of organelle movement? And you would tell me microtubules. I might ask you, what's in charge of changing cell shape? And you would tell me microfilaments. Or I might ask you, what anchors the nucleus in place? And you would tell me intermediate filaments. So make sure that you study this table very well. Remember, cell wall is made out of cellulose. We talked about in chapter 5, and it has 3 functions. Know those functions, and that these are the channels that cells use to communicate with each other. The extracellular matrix is the area between cells. And remember, it has three functions: support, cushioning, and communication. Made of glycoproteins. Remember, these are proteins with sugars attached to them. All right, so that covers chapter 6. Let me pull up chapter 7. Okay, so chapter 7 has to do with the membrane. We talked about membrane structure. It's a phospholipid bilayer that it's very thin, and it controls what goes in and out of the cell. It's selectively permeable, so some objects can cross and others can't. We talked about the word apopathic. Make sure you guys know that. That means it has a dual nature. In this case, phospholipids, one end of them is water loving, and the other end is water fearing. And you know, why is that important? Well, it's important because you couldn't have a cell that had a membrane that dissolved in water, just like you would want to build your house in something that was water soluble, because the first time it encountered water, all the walls would dissolve, and we couldn't maintain homeostasis, which would result in death. We talked about that in chapter 1. Fluid mosaic model just says that the phospholipid bilayer is like a sea, and the proteins can float around within it. So nothing is fixed in any position. You don't have to know freeze fractures, so skip that. We talked about how we know, and that the fluidity of the membrane is temperature dependent. So make sure you know that. We can change the fluidity of the membrane based on the temperature that the cells encounter. So, you know, your skin cells may be at a different temperature than, say, your heart cells. So you need to be able to manage this, because if it gets too cold, the channels will squeeze shut and you can't pass things through into the cell. And if it's too hot, the membrane would sort of turn into goo, and you don't want that. So to do that, plants change their fatty acid tails from saturated to unsaturated, and that makes it more fluid. But we don't have saturated and unsaturated fatty acid tails. And if you're not sure about that, go back and look at chapter 5, when we talked about the difference between saturated and unsaturated, and where saturated fats usually come from and unsaturated fats usually come from. So in animals, we use cholesterol. Cholesterol is a very important molecule, not only to synthesize steroid hormones, but to maintain fluidity in the membrane. And you would expect things that live in cold conditions would have more cholesterol, because it's physically bending these than things that lived in warmer environments. All right, so remember that the consistency of the cell membrane has to be about the same consistency as vegetable oil. And if that changes, then the cell is screwed, because it can't bring things in. It can't maintain its relatively constant internal environment. We talked about integral and peripheral proteins. Remember, the integral ones go across the membrane, peripheral ones are on the periphery, like your peripheral vision. They have different functions, so make sure you know the functions. We talked about that things can pass through the phospholipid bilayer without a channel, and the rule is those are small and hydrophobic and electrically neutral. So make sure you guys know that. Know the rules of diffusion: high concentration to low concentration, continues until everything is equal. Osmosis is a special condition where that just involves water. Remember, the cells have these constantly open channels called aquaporins, and that water can flow freely in and out of the cells, but salts and other things can't, remember, because they're charged, and they need channels to be made for them. They're not always open. So hypertonic, generally, because the inside of the cell is constant, a constant tonicity, salinity, or a concentration of ions, we refer to these outside of the cell, but sometimes that can change, so make sure you pay attention when you're reading it. So hypertonic means that it's high salt outside of the cell. Hypotonic means that it's low, so hypo is low outside of the cell. Isotonic means it's the same. And this is how cells like to be, like animal cells like to be isotonic. Plant cells like to have a little water because that gives them pressure against them. So they like to be in a hypertonic solution. Remember, water goes in, in a hypotonic solution, because remember, water runs in a hypotonic and runs out in a hypertonic. So animal cells in hypotonic fill with water and they burst. And hypertonic, they shrink. And remember, you have to have a certain amount of room to work. So too small cells don't function. And then isotonic solution is really like how animal cells like to be. That's different. Plant cells like to be in a hypotonic solution. Hypertonic solutions cause their membranes to rip apart and they die. So it's not good. Come on. Why is it 2 slides? Okay, so we have facilitated diffusion. That's when we have a channel and things go from high to low. Remember, some things can pass through the membrane, but they have to be small, uncharged, and hydrophobic. We can have gated diffusion, so it goes, remember, when one molecule binds to another, it changes its shape. So that's why it's open on the outside here. When it binds, it's closed on the outside. Active transport requires energy. If you want water to run uphill, you have to put energy in it. If it's doing it on its own, it's passive. We talked about the sodium-potassium pump. This is an example of active transport, and this is how we build up. We charge our muscles with electricity by 3 sodiums coming out, 2 potassiums going in. That's an imbalance in positive charges. So positive charges on the outside, negative on the inside. And you guys know, by hooking up electricity to your muscles, you ever been to physical therapy or whatever, it will cause your muscles to contract. So this is a summary slide. Diffusion across the phospholipid bilayer is possible, but only for small uncharged hydrophobic molecules. Facilitated diffusion is more common. This is how like glucose gets in from your blood into your cell to do glycolysis. And then honestly, a lot of times you do active transport where you have to go from a low concentration to high, and that requires energy. And remember, your energy currency is ATP. These are terms you need to know: membrane potential, You don't need to know specifically what the voltage is, but you should know that usually the positive charges end up on the outside, negative charges on the inside. So cations and anions, you should have learned that term in chapter 2. We call this an electrochemical gradient because it's an electrical gradient, you know, high on one side, positive charge is low on the other. That's produced by chemicals like sodium and potassium. And that can be used to do work just like a battery. So we call this an electrogenic pump because it's generating electricity by pumping or through active transport. And we talked about sodium, potassium, but we can also do this with protons. And this is how the mitochondria does it. And bacteria. Co-transport is when you transport something downstream actively and then you bring it in passively. Exocytosis is things going outside of the cell in large amounts, like not just a channel, but a whole bunch, like your saliva or milk in mammary glands. Endocytosis, there's 3 kinds, phago, pino, and receptor-mediated endocytosis. This is food, this is drink, and this is special molecules like low-density lipoprotein, the bad cholesterol. And this is how things are brought into the cell. So make sure you guys know all that, know what pseudopodia means, know what phagocytosis is, know what pinocytosis is, know an example of receptor-mediated endocytosis, like we said, LDL. And that's it for chapter 7.
I don't know how much to take down on the wall. Yeah, you just want to.
So I'm gonna end this here, and then I'll pick up and do chapter 8 and 9 when it's not so loud. So this is the review for chapters 6 and 7.